Does life's molecular replication apparatus evolve from a common ancestor? We can test this hypothesis by looking across the genomes of various species for recognizable sequence similarity in the genes that code for human helicase. It's a 6-step process. We'll begin by identifying the cellular process involved using the KEGG database. Let's search for DNA replication as the process. We find that there's a KEGG pathway. This pathway is described as being the replication pathway, and it has associated with it a pathway map. Let's go there. Study the map and you'll find that there are prokaryotic as well as eukaryotic processes. The two daughter strands coming from the parent strand can be found. And what we need to do next in our workflow is find the relevant protein that's associated with helicase. There it is at the fork. There's going to be MCM2 through 7 components, protein components that make up helicase, and we can see them in the box at the right. Either of these, any of these, can be clicked on, and it'll take you to the DNA replication licensing factor MCM2, and so that's our relevant protein. And now we need to find the gene for the human MCM2. If you scroll down, there it is. HSA is human, the Homo sapien, and the Homo sapiens MCM2 gene can be found in the KEGG pathway. And if you scroll down, you'll see the nucleotide sequence, all 2715 bases. Now we want to search the genome of various species to see if they have a gene that's similar to human MCM2. We'll do this search with a tool known as BLAST, and we'll use nucleotide BLAST or BLAST-N, and we'll search against first the mouse. So we're looking in the mouse genome to see whether there's a gene that's similar to that human MCM2. And now we've done the search and we want to compare and analyze the results. The score is high. We found a gene, it's MCM2, with a high score and an expect value, a probability of having that alignment by random chance of near 0. You can see by looking across the alignment that there's a gap there, there's a gap there, and there's a few inconsistencies in the nucleotides, but overall, across that gene, there's almost very good, perfect match. Now let's look for the chicken and see whether we can find a similar MCM gene with the species GGA. We do. We find an MCM2 gene, a score a little bit lower, but still an expect value of 0. The chances of getting this good of alignment by random chance is near 0. Again, across that whole gene, we can see good agreement. And let's look now at how well that agreement matches across the whole gene. Our query is human, our result is the chicken, and you can see that nearly across that whole human gene, there's a high value, a high bit score. Finally, let's go ahead and look for budding yeast, something that's quite far removed ancestrally from humans. Again, we find an MCM2 gene, but now you'll notice that the score is quite a bit lower, and the probability has risen from something that's non-zero. It's 1 times 10 to the minus 26. And here you can see that the alignment is really good, only across a small portion of the human gene. Here are our results. This preliminary evidence suggests that there is indeed a common origin of the DNA replication apparatus.